Our next talk is Natalia Komarova, dialing in remotely from UC San Diego, who is going to continue talking. Has been a longtime collaborator with John, even when the ancient, bug and whippy, buggy and whip days when I was a graduate student at UC Irvine. So we're really thrilled that she was able to join us for a talk here today. Yeah, if you can please full screen, share your screen. Yeah, perfect. You can go to presentation mode if you can. That is perfect. Thank you.
Thank you so much for inviting me. Happy to be here. My talk will be about a connection between a microscopic description of a cancer and an epidemiological scale where we are dealing with populations of people. So mathematics can provide a connection between these two, which I always found curious. But before I begin, I wanted to show you a picture of my wedding. So you see this beautiful couple here. This is not a bride and a groom, but I think John may recognize who these people are. Such a cute couple. So I've been looking through the old photos. Those were taken even before the advance of digital photography. So, and I became all sentimental looking at these people. Look at them pulling silly faces in the next shot. And I have another one. Most of them are John's. And they're very, very young here. And look at these two best friends. So at the beginning of my talk, I just wanted to congratulate John not only on the beautiful career that he has, but also on everything else. And I'm very proud and honored and happy to be his friend. So back to this. We'll talk about the role of aspirin in kind of preventing colorectal cancer. So this is an important, colorectal cancer is obviously an important problem. It's the third leading cause of cancer-related deaths in the United States for both men and women. So hundreds of thousands of new cases come up every year and tens of thousands of deaths occur. So it's usually a condition that comes sort of later in life, but I will talk about other possible conditions. So it affects more developed nations, possibly because of lifestyle factors, such as diet, a lack of physical activity, and higher life expectancy. So we model colon cancer by looking at the place where it starts, which is colonic crypts. These are these little folds of skin that are observed in colon. They're microscopic, about 10 to the 3, 10 to the 4 cells per crypt. This is the horizontal, this is kind of a sideways projection of a crypt with stem cells residing here at the bottom. And this here on the right is a commonly mapped progression from normal epithelium to colon cancer, which includes several, up to 7 different steps that have been mapped genetically. One of the earlier events is an inactivation of the APC gene here. So it kind of starts with the APC gene present in both copies, and one of them gets inactivated, then the other one, and then so on, it goes on. So related to that, there is a familial condition where a person is born with one copy of this gene already inactivated. So kind of this first step is taken away. And this first step, in fact, is the longest in the in this progression. It takes tens of years, dozens of years. So if this step is no longer there, the progression is accelerated. So people with this condition called FAP get colon cancer in their late teens. Another way in which a person can be predisposed to colorectal cancer is this. There's a parallel pathway to cancer where as a first step, one of the MSI genes is inactivated. So we're talking about microsatellite instability. If those genes are inactivated, cells make more mutations, a certain kind of mutations. So once this gene is inactivated, everything else goes faster. So first 2 slow steps and then very fast steps. So there are people who are born with these genes, with one of the copies of these genes inactivated, which makes the acquisition of microsatellite instability faster, and then it progresses faster from here. This is called the Lynch syndrome. And colon cancer and also other cancers are acquired by the age of about 50 or so in this cohort of patients. So I'll be mentioning this in this talk. So I will start actually with the right-hand side of this picture, where we talk about the epidemiology of cancer and how aspirin can affect that. And so this first study that I want to describe was conducted by John Byrne in Great Britain. It was called CAP2 study, a very large clinical trial where they looked at the effect of aspirin in the cohort of Lynch syndrome patients. So again, Lynch syndrome is the one I just described. These are people who are predisposed to colorectal cancer and usually get a cancer in their 50s or 60s. So for these patients, getting cancer is a much more likely occurrence. So a large number of these patients were given either a 600 milligram aspirin dose or aspirin placebo, and the results were compared over many years. So it was a very long study. So here in blue is aspirin placebo, and in red is aspirin. And we're looking at the proportion of the patients diagnosed with Lynch syndrome cancer, any of those cancers. And you can see that after 11 years of this trial, there's a big difference between those who took aspirin and those who didn't. So the incidence of cancer has been significantly reduced. And this is another plot from a similar study. There were other many, in fact, studies that looked at this. So this is kind of like a meta study that summarizes their results. We're talking about risk ratio. Risk ratio is defined as the probability to get cancer with aspirin divided by that without. So if this quantity is less than 1, it means that aspirin helps. This vertical line is 1, and we're plotting risk ratio that was obtained in all of these different studies. And you can see that everywhere. For all these studies, the risk ratio is smaller than 1, which means that somehow aspirin reduces the probability to get colorectal cancer. But this is the study that's most relevant for us. This is a study of Andrew Chan. And that's not a clinical trial. It's rather a questionnaire-based study that was conducted among 10,000 nurses. So all the nurses were asked how many aspirin tablets they took per week. And the answers ranged from 0 to more than 14. So 14 tablets per week means 2 tablets per day. So this is kind of like a standard dose you take for headache or so. And so they looked at the prevalence of cancer. And look at this. For the largest dose, the risk ratio was about a half, which means that for these people, the probability to get colorectal adenoma, that's so they were focusing on colorectal adenoma, it was reduced by a factor of 2. So we wanted to look more into this and kind of see how that may work. And this is a collaborative project with two experimental scientists, Ajay Goel and Rick Boland. And it was done in collaboration with Dominic Wodards and our former postdoc, Yifan Wang. So we started with our collaborators were doing experiments in vivo and in vitro to look at the effect of aspirin on cancer cell lines. So they injected aspirin into mice that had xenografts of these tumors growing, and they looked at the growth curves of tumors. So these are some of the photographs from the study for different cell lines and for different doses of aspirin that they administered. Visually, you can see here that, so these are xenografts for 0 aspirin, and this is the largest dose of aspirin. You can see they're smaller. So let's take a look at growth curves. As a function of days, here we are plotting the experimentally obtained data on tumor volume in millimeter cubed for different doses of aspirin. It kind of goes down. So, and these 4 graphs correspond to for different cell lines. So we looked at the surface growth equation and fitted these data to obtain division and death rate of cells for different cell lines and for different doses of aspirin. And we did the same thing in vivo. I'm sorry, this is in vitro. And the results are very similar. So here is the main result. As a function of the aspirin dose, we're plotting the death rate of cells that we measured and the division rate of cells measured. And you can see that as the dose increases, the death rate increases and the division rate decreases. And exactly the same thing was also obtained in vitro, but the model there was different. So things grow exponentially in vitro, and these grow as t cubed in vivo. So we wanted to use these data to see how this reduction in the division rate and the increase in death rate effect possibly can be translated into the epidemiological data. So this is an age incidence curve which was taken in the absence of aspirin, just for regular patients. This is the incidence of advanced adenoma as a function of years. And here the blue and red correspond to the 2 genders, which we didn't know how to take that into account, so we took the average. So let's look at the black. So this is the black curve. And we know that under aspirin, with different doses, this curve gets somehow lowered. And for the largest dose, it's lowered by a factor of 2. So the question is, why? So we created and parameterized the model of late adenoma generation. We implemented the effect of aspirin through modification of kinetic parameters. And this is the degree of this modification was measured by us, by our collaborators. And we connected that with epidemiological population data and measured the reduction which resulted from the aspirin. So let's take a look. Back to the microscopic part of the problem. We want to understand how this progression goes. And up to late adenoma, there are 2 mutations that are of importance. One is the inactivation of the APC gene, the tumor suppressor gene, and the activation of the oncogene KRAS. So we look at one compartment, one crypt, right? And we look at the rate of mutations and how these mutations spread, and the next mutation and spread, right? There's a lot of theory that goes into this and that we have developed over the years. But this is how the picture looks like if we take into account the genes that are important for late adenoma. APC gene gets first copy inactivated, second copy inactivated, and the oncogene is activated here. So this is advanced adenoma. Or the oncogene can be activated first and APC inactivated second. Or you can go like this, right? So we can also take into account this weird phenomenon of crypt fission that became discovered relatively recently, where mutated crypts somehow multiply and expand. And that's an important factor when we talk about the progression. So this is a system of equations that was taken from this paper. And we, for the first time, we were able to apply parameter values that were recently measured for many of these rates. So that's what made this study possible at all. We tried it for many years before, and there was just too much uncertainty in the parameter ranges. But now this is something that one can do mathematically because experimental science has advanced so much that we know a lot of the rates. We modified the model to include saturation growth because things don't grow exponentially. And we first wanted to kind of calibrate the model because even though I said most of the parameters are known, not all of them are known, so we had to fit them. So instead of just one set of parameters, we had ranges of parameters where fits are the best. So whatever results we got, we apply to these ranges. And so how do we incorporate aspirin? We assume that from birth, time goes by, and at some point the aspirin starts being taken, and maybe the treatment lasts for a certain number of years. And then there's a measurement, follow-up time, when we can calculate the relative risk of acquiring late adenoma. And we want to use the parameter values that we measured, but of course the measurements were done in mice. To our delight, there exist tables that allow one to translate things from mice to humans. So whatever dose was used in mice can be translated into, tablets per day in humans. And the effects of aspirin are twofold. The first type of effect is intracrypt dynamics. So inside each crypt, these transitions are affected by the reduction in the division rate and an increase in death rate of cells due to aspirin. And we can calculate how they are affected because we can calculate all these rates. The second way the equations are affected is through these crypt fission rates, which are here. These crypt fission rates are related to cell division, so obviously this parameter is also affected. And here is, we are looking at the first set of results. Let's take a look. So each of these graphs corresponds to our simulations, where we administer, in silico, administer aspirin for 1 decade of a human life. So here from the age of 20 to 30, here from the age of 30 to 40, and so on, until 60 to 70, 70 to 80. The black dots, thick black dots, are the epidemiological data in the absence of aspirin, and the thin black line is our model fitted to the data in the absence of aspirin. So in the absence of aspirin, the age incidence curve goes through the epidemiological data. That's good. So now let's in silico apply aspirin. And if we apply aspirin between 20 and 30, that means that the model parameters during that time are changed in the systematic way that we measured. And this leads to a reduction in risk, which is plotted by the yellow line. So if this person, if this in silico person took aspirin between the ages of 20 and 30, this is the risk reduction that they would experience. Okay, and so you can see that the risk of cancer is reduced even if the treatment happens later, even if it happens much later. The reduction in risk is not as large, but it's still there, which is good. So this is where we study by how much the risk is reduced depending on the dose and also depending on the age when the aspirin is taken. So strong dose means 2 tablets per week, and light dose is the lightest dose that we had in the nurse study by Andrew Chan. And for strong dose, you can see that the reduction of risk is compatible with what they measured. You see this here, about 50%. And then the later the treatment happens, the less is risk reduction. But it's always, you see, noticeable. And it also depends on the dose in this way. We can look at the dependence of the effect on the length of treatment. So we can compare a 10-year treatment with a 5-year treatment. Here, yellow is 10 years and blue is 5 years. You can see it kind of splits it in half almost like this. And we also can play with the model and see which types of cells are affected by aspirin. So these studies are possible in silico, but very difficult to get this information from even a very large questionnaire, right? Because once you start splitting the group of patients into different classes, like how long did they take aspirin for? What was the age when they started? What was their dose? The more you split, the less statistical significance you get. So a mathematical model like this that was calibrated to the data can do this job and kind of make all the predictions and kind of reconcile the data with what we know. So our previous work indicated that aspirin reduces the rate of cell division and increases the rate of cell death in a dose-dependent way. So we measured that, we stick that in the model, and we showed that with these parameter changes, the risk of colorectal adenoma was reduced by about 50% in the case of the strongest dose, which is consistent with what was observed in a study. So that is the take-home message here. And I want to thank my group, my collaborators, and thank you for listening.
Thank you very much for a wonderful talk. I already